Hello, this is Dr. Michael Edwards, founder and CEO of Bioinfo Solutions, a data analytics company based out of Parker, Colorado. This entry is part 4 in a series of instructional videos detailing a meta-analysis on 8 different human gene expression studies looking at papillary renal cell carcinoma. In this installment, I use Partec Genomics Suite to display the metadata as a heat map and employ hierarchical clustering to find connected genes in a dataset based on its normalized gene scores across all 17 different comparisons. A text file containing the papillary meta-analysis used in this video can be accessed at a Google Drive link provided in the description of this YouTube entry. In our previous series of videos, we had collected all the Illumina normalized scores for the top 5,000 genes differentially expressed in our meta-analysis. Again, these top 5,000 genes are based on the sum of the absolute value of these gene scores. We also formatted the data to reflect the type of change that occurs in that particular comparison. So that in this case, this gene went up since it has a positive value in this comparison, while this particular gene went down. Again, the scale based on our formatting is from negative 100 to a positive 100. We then copied and pasted all of the Illumina gene scores for the top 5,000 into Partec's Genomics Suite. After we have pasted it in, we then merged a file that contains all the experimental and clinical data that we could find on these comparisons. Once we have attached all this clinical information, we could then use principal component analysis to visualize this data in three-dimensional space. And from this visualization, we can kind of get a sense of how we expect our particular variables, the types of different cancers, how is it going to affect the gene expression. So what I'm going to show you today is another popular visualization method, and these are called heat maps. So what we were going to do is take all of this Illumina scaled data and turn it into different intensities of color on a graph. Not only will we be able to visualize the genes, but we will group them based on similar patterns across all these different comparisons, with the thought being that things that behave similarly across many different studies are probably acting together. And so what we're going to do is use the clustering to try to pull out biological information out of this data set.